Okay, everyone, we're going to go over the virtual Mitosis Lab. So in this lab, you were supposed to click on this link, and when you did that, it brought you to the virtual microscope. You clicked on Explore, you clicked on the slide case, you opened the slides, plant and animal cell slides, and you then selected different various plant or animal cell slides, and you viewed them. So in this case, I pulled up one of the animal cell whitefish mitosis slides that are in Metaphase. So in this case, I'm looking at cells that were in Metaphase. So on your lab, you're supposed to do something similar to this. You were supposed to make a drawing of the phase, include the correct total magnification, try to identify how many cells were in that phase, and then identify the phase and write a description of the phase. And this is an example of a real slide, a picture of a real slide of cells in interphase. This one happens to be the end of a root tip, onion root tip. So here's a student example of what that should look like. So the directions tell you 2 plant cells. There's one here, there's one down below, and 2 animal cells. There's one here and there's one down below. Okay, they identified they drew a picture, identified things on their picture so we know what they're talking about, included a total magnification, included the number of cells in the phase, identified the phase, and wrote their description. In this case, they said prophase, and they said that prophase, the nucleus is starting to break down. I think that's good, showing the nucleus spreading out and breaking down. I added some information in green throughout the lab here. So spindle fibers form, and they attach to the middle of the sister chromatids, which is not in picture, but that's okay. At the middle of the chromosome is called the centromere. The centrioles are moving to opposite ends. All of these other things happen during prophase. Okay? In interphase, they wrote a cell in interphase, identified it, and they wrote that the nucleus is still together, so that means it's in interphase, the spindles are still forming. In addition, it's important to remember that genetic material gets copied at the end of interphase, and the centrioles are starting to move to opposite ends, preparing for the first stage of mitosis and cell division. Okay? Let's talk about the correct magnifications. This was something I looked at in your lab, and I think you needed to have at least 400x in order to get credit for it, and this is why. This is 400 times 40 times the ocular lens of 10, gives you a total magnification of 400 times. I think that if you put 4x, let's go in order. If you put 100x, which would be 10 times 10, it's too small to really see. And certainly 4x was really small, right? 4 times 10 is 40x. 10 times 10 is 100x. These are too small to really identify the stages. So if you didn't at least put 400x, 40 times the ocular lens of 10, 400x for your total magnification, you couldn't get proper credit. Now here's a cell in interphase, and the idea was to try to count how many cells. So as you scroll through the slide, I'm looking for cells that look like that one. There's 2. Okay, and you click on the screen and you simply go through the slide. And this is really what it would be like. You would be physically moving the slide around the stage trying to find other cells. So that's all I'm doing here. That's what this simulates. And I only count 2 cells in metaphase. 2 cells where the chromosomes are lined up down the middle of the center of the cell. I don't know if that was the original one, but 2 to 3 is what I would put on my lab. Now, in the student example, you can see they're way off, but that's not the point. The point was that you were trying to preview the slide and do a compare and contrast kind of a thing and identify the different stages. Here are the other 2 drawings on the next page. Again, nicely labeled, nicely drawn, correct magnifications. Tried to identify the number of cells. This one's anaphase. The chromosomes separate and start to pull toward the poles. And by the poles, we mean the centrioles. And I don't think there was anything else to add there. That's perfect. And then metaphase was one of your required drawings. And the chromosomes are attached to the metaplate and the mitotic spindle fibers as well. Metaplate's not really the correct name. It's the not the metaplate, but the metaphase plate. That's the center region. And then the distinctive characteristics, the copied chromosomes, the X's, right, are 2 attached chromosomes. They are getting ready to be pulled apart in anaphase over here. And so they're attached at the middle of the chromosomes, at the centromere, and each copy, these sister chromatids, sisters, are connected as X's, but are going to be pulled apart in the next phase so that each cell can get a copy of all of those chromosomes. Now we're on to the analysis questions. And what is mitosis? Why must it occur? It's a stage in the cell cycle where the cell divides. It's important because it creates new cells. It also helps in repair replacement of worn out cells. Okay, and I could have really added that mitosis distinctly is about getting two new nuclei so that you can get a nuclei with all of the genetic information into the two new cells. Mitosis stages: interphase, prophase, metaphase, anaphase, telophase. They did add cytokinesis, which I explained here is not a part of cell division. Mitosis. It is a part of cell division, but mitosis ends at telophase, which is the creation of the two new nuclei. We then have to divide up the cytoplasm and the organelles, and the cells have to pinch, so we get two new cells. Interphase is an important stage of the cell cycle. It grows. It also carries out specialized functions, right? So if you're a blood cell, you're carrying oxygen to cells, carbon dioxide away. If you're a muscle cell, you're helping to move bones. If you're a nerve cell, you're carrying signals around the body, those kinds of specialized functions. Plant and animal cell mitosis, the difference is plant cells do not have centrioles, where animal cells do use centrioles. That was fine. That was good enough. I did add that organelles similar to centrioles in plants are called centrosomes. And the cell also has this mid-region called the cell plate, which is a way how the cell wall divides during their cell division. Middle phase is metaphase, where we've got the chromosomes lined up in the middle. And mitosis, why is there a lot of mitosis in whitefish egg cells? Because you start out as one fertilized egg, sperm unite, that's fertilization. All the genetic material is now in a single cell, and now this cell wants to grow into a multicellular organism like a fish. Well, it's got to keep dividing and going through mitosis many, many times and adding lots and lots of cells. Why is there a lot in the root tip? It's not really growing to form an organism. A root tip is just a specialized part of a plant, the root, and the root tip is where the end of the root is. And roots are constantly growing to find water, either out or downward, and because plants need water, it's so important for photosynthesis, and so that's why there's a lot of growth there. Okay, so if you were missing some of these things, you lost points in your lab. Obviously, the more you missed, the more your grade was reduced from 100. This was a really good lab, even though there were a few problems with the number of cells that they identified. I thought it was a good effort, and so this lab was 100.